The advances in scientific research have made medicine evolve greatly in the last decades. Treatments have gone from being generalized to become more and more precise, and hence their effectiveness has greatly increased. Since the discovery of the DNA structure in 1953 that revolutionized our understanding of biology, researchers have deepened their knowledge on the molecular mechanisms of life to use this information to fight diseases. To date, we know that our body is a combination of billions of cells, and each of them carries within its nucleus a set of our chromosomes made of DNA with all the essential information for our body functions. The DNA molecule is composed of two chains forming a double helix with a sequence of 4 base pairs: adenine, guanine, cytosine, and thymine. And at the same time, it's organized in up to 20,000 units known as genes that contain the instructions for the cell. But these genes never leave the nucleus. So, when the information is needed, it is transcribed into another molecule called messenger RNA. This one can leave the nucleus and carry the information to the rest of the cell. To do so, the messenger RNA has to be interpreted by the ribosomes and translated into proteins, the essential bricks that finally execute the vital functions of our cells. In the lab, we can culture cells in incubators to later extract and analyze their genetic material. It is possible to read the sequence of the DNA, providing us with the entire genome of a single cell, or even of a complete organism. Given that this code and process is conserved across species, we've been able to study it in deep, and so the mechanisms that regulate it. Organisms like plants, for example, have shed light on an interesting molecular mechanism to disrupt protein production. In the early 90s, researchers from the United States discovered that during this process, small pieces of RNA could bind to the messenger RNA, degrade it, and thus block the synthesis of the protein. Later, Mello and Fire discovered the structure of these RNAs and called the process RNA interference. They saw that this mechanism could be a way to disrupt protein production by switching on and off the expression of genes. By mimicking what happens in nature in the lab, we can design RNA sequences that are capable of silencing genes of interest. We call these molecules small interference RNAs, or siRNAs. Once we have designed them on the computer, we can synthesize them and use them on cells to verify if the translation of the gene is blocked, and thus the protein is no longer translated. A feature that makes siRNA a great ally to treat pathologies. For example, in conditions such as inflammatory bowel diseases, some cells of the gut produce high levels of pro-inflammatory proteins causing the inflammation of the bowel. In this context, siRNA could become a treatment, silencing the genes that express these proteins and thus reducing the inflammation. In recent years, there has been a lot of discussion about the high potential of these molecules to become treatments. One advantage is their high specificity that reduces the side effects considerably. This is important when we're talking about chronic diseases that need prolonged treatments. Research is focusing on the design of the best siRNA molecules to target specific genes and cells. We also need to consider that to have an effect, the siRNA must arrive to those cells. These molecules are very sensitive and easily degradable, and thus we have to figure out a way to make them reach their destination successfully. The same way we put drugs inside a pill and pills inside a box so that they reach the consumer in perfect condition, we have to pack the siRNAs until their finish line. To do this, we use lipidic nanocarriers. These are tiny fat-made spheres where our molecules can be embedded. The lipid nature of the nanoparticles allows them to trespass the cell membranes and to release the siRNAs inside them, where they can block the messengers. In the lab, we can produce these nanoparticles mixing the lipidic ingredients to create an emulsion and obtain a suspension that contains millions of nanocarriers. These spheres are highly stable. Their production process is controlled and can be easily scaled up. Once we have produced them, we must ensure that the composition and the size of every single particle is uniform to avoid any toxicity. From the first experiment to the last one, the daily work of researchers brings the great potential of siRNA and nanoparticles a step closer to the clinic. If you want to know more about nanotherapy research against inflammatory bowel diseases, go to www.newdeal-project.eu.